setting in Denver, Colorado, labs in garage and basement, graduated high school 2 years early, University of Chicago chemistry. सर ये skip कर दो उनका सब ये like हेलो हाँ uh, Woods 1954 Masons went to Woods Hall to be a teaching assistant. Sattel was a postdoctoral fellow at the Wood Hall where he was studying molecular biology techniques. Throughout the summer, Masons and Sattel and discussion on theory experiment shared similar ideas were keen to work together. Sattel received a postdoctoral position in Catholic, particular interest in building of a Watson and Crick DNA model. The DNA model, Watson and Crick DNA model, 1953. Double-stranded helix, right-handed nucleotide base from interior sugar phosphate backbone, aligned with Cherof's rule, each purine paired with a pyrimidine ATGC sequence. Watson and Crick speculated that the specific base pairing were indicator of Paris mode of DNA replication. So, there is three major hypotheses for DNA replication. So, the semi-conservative direction. In this model, the two strands of DNA unwind from each other, and it acts as a template for synthesis of a new complementary strand. This results in two DNA DNA molecule with original strands and DNA. So एक मिनट सर just a second. हाँ सर एक मिनट जस्ट मैं बाद में देते सर
प्रतीक नहीं है
यस सर हाँ सर चलेगा हेलो सर मैं आ गई सर हाँ सर शेयर करूँ स्क्रीन ओके सर हेलो सर आई गेस कुछ इश्यू हो रहा है, मैं आपको सेंड करूँ? ओके सर, मैम। ओके, ओके। So good evening, everyone. My name is Sakshi, and today I'm gonna represent eukaryotic DNA replication. So basically, eukaryotic DNA replication is a conserved mechanism that restricts DNA replication to once per cell cycle. Eukaryotic DNA replication of chromosomal DNA is essential for duplication of a cell and is necessary for maintenance of eukaryotic genome. DNA replication is an action of DNA polymerase synthesizing a DNA strand complementary to the original template strand. Enzyme responsible for replication, helicase. Helicase is an enzyme which unzips the DNA strand by breaking the hydrogen bond between them. Thus, it helps in the formation of replication fork. A protein called single strand binding protein coat the separate strands of DNA near the replication fork, keeping them from coming back together in a double helix. So the another enzyme responsible in eukaryotic replication is DNA polymerase, which is divided into DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase epsilon. As you can see, polymerase activity of these DNA polymerase is from 5 prime to 3 prime. DNA polymerase alpha shows replication and DNA repair, while DNA polymerase beta shows DNA repair, and DNA polymerase gamma shows replication in the mitochondria, and DNA polymerase delta show replication and DNA repair, while DNA polymerase epsilon shows replication. Out of these DNA polymerase, uh, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase epsilon shows the 3 prime to 5 prime exonuclease activity, while the DNA polymerase alpha and DNA polymerase beta does not show these activities. As I have explained, the same is written over here. DNA polymerase alpha and DNA polymerase delta replicate chromosomal DNA. DNA polymerase beta and epsilon repair DNA, and DNA polymerase gamma replicates mitochondrial DNA. DNA polymerase alpha and delta synthesize the lagging strand via Okazaki fragments. The RNA primers are synthesized by DNA polymerase alpha, which carries a primase subunit. DNA polymerase delta synthesizes the leading strand. Telomerase, a DNA polymerase that contains an integral RNA that acts as its own primer, is used to replicate DNA at the end of the chromosomal, that is telomeres. DNA topoisomerase 1. It relaxes the DNA helix during the replication through creation of a nick in one of the DNA strands. DNA topoisomerase 2. It relieves the strain on the DNA helix during the replication by forming supercoil in the helix through the creation of nick in both strands of DNA. And the another enzyme is DNA ligase. It forms 3 prime to 5 prime phosphodiester bond between adjacent Okazaki fragments of DNA. Thank you. Hello. Sir, DNA polymerase 1. DNA polymerase alpha and beta. No, sir. I don't know. Once again, can you repeat question? Sir, DNA polymerase delta. Sir, formation of nucleotides. No, no, sir. It was enzymes responsible for eukaryotic replication. Sir, it was just enzymes. Okay, thank you so much, sir.
सर मेरा लो ऋतुजा सर या या आपको सेंड किया है यस सर गुड इवनिंग गुड इवनिंग dear sir and dear students. My name is ऋतुजा Jagannath Yadav. I am going to speak about topic replicons and initiation of replication in eukaryotes. Okay. So first of all, we will see what is replicon. The DNA replicate form a single origin. It's called a replicon or replication unit. Eukaryotic chromosomes generally contain much more DNA than those of prokaryotes, and their replication forks move much more slowly. If they were likely typical prokaryotes with only one origin of replication per chromosome, DNA replication would take many days. Instead, eukaryotic chromosomes contain multiple origins at which DNA denatures and replication then proceeds bidirectionally until an adjacent replication fork is encountered. In eukaryotes, replicon size is smaller than it is in prokaryotes. Replication is replication is slower, and each chromosome contains more replicons. Number and size of replicons are vary with cell type. Not all origins with a genome initiate DNA synthesis. सर मैं आगे अब बोल रही हूँ simultaneously, cell-specific patterns of origin activation are observed so that chromosomal regions are replicated in predictable order in each cell cycle. So initiation of replication. Eukaryotic origins are generally not well characterized. Those of the East Saccharomyces cerevisiae are among the best understood. Chromosomal DNA fragments, about 100 base pair, that are able to replicate autonomously when introduced into yeast as extracellular. Circular DNA are known as ARS. ARS are yeast replicators. The three sequence elements typically found in ARS are A, B1, and B2. Initiator protein in yeast in the multiprotein origin recognize complex which binds to A and B1. Other replication proteins join include only one that unbinds DNA and B2. The yeast origin of replication is between region B1 and B2. DNA and histones must be doubled in each cell cycle. G1 prepares the cell for DNA replication. Chromosome duplication occurs during S phase. G2 prepares for cell division, and segregation of progeny chromosome occurs during M phase, allowing the cell to divide. Sir, cell cycle control is complex and only outlined here. Each in which chromosomal replication is well studied, several uh, so as a eukaryotic model organism. Initiation of replication has two separate steps controlled by cyclin-dependent kinases that are present throughout the cell cycle except during G1. In the absence of CDSK during G1, replicator selection occurs. ORC and other proteins assembled on each replicator to form pre-replicative complexes. When cell enters S phase, CDKs are present and active pre-RS cell to initiate replication. CDK active inhibits another round of pre-RC formation until the cell again enters G1 when CDK is absent. Thank you. Yes, sir. In which divides. Sorry, sir. Once again. Stage na, sir. M phase divides and out in S phase. Uh, cycling activity inhibits another round of. Um, sir, I don't know. How many replicons are in eukaryotes? Uh, 1,000. 10 to 330 best pair. 10 to 330 best pair. Kilo base. So I don't know. Kilo best pair. ORC. Origin recognition complex. Active pre-RC formation until the cell again enter G1 phase. CDKs present throughout the cell cycle except G1 and this much, sir. Regulating cell cycle. Sir, nice, sir. एक साथ सेम टाइम पे नहीं होते सर रेप्लिकेट इंटरफेस एक सेकेंड सर इंटरफेस नेम थैंक यू सर
सर मैं रेडी हूँ. Yes, sir. Hello, sir. Hello, everyone. Today I'm going to represent Mason and Sattel experiment. Sir, मैं direct start कर रही हूँ. Hello, sir. हाँ. So, हाँ. So there is a so there were three basic models of DNA replication that had been proposed by the scientific community after the discovery of DNA structure. So as you can see, semi-conservative replication. In this model, the two strands of DNA unwind from each other and each acts as a template for synthesis of new complementary strands. This results in two DNA molecules with original strand and one new strand. Thus, then a conservative replication. Uh, in this model, DNA replication results in one molecule that consists of both original DNA strands and another molecule that consists of two new strands, sequence as the original. Uh, uh, sorry. And next is dispersive replication. In this dispersive model, DNA replication results in two DNA molecules that are mixture or hybrid or parental and daughter DNA. In this model, each individual strand is a patchwork of original and new DNA. As you can see in this diagram, they explain the explain experiment. They big Mason and Sattel conducted their famous experiment on DNA replication using E. coli bacteria as model system. They began by growing E. coli in medium or nutrient growth containing a heavy isotope of nitrogen 15. When grow on medium containing heavy 15N, the bacteria took up the nitrogen and used it to synthesize new biological molecule, including DNA, uh, including uh, including DNA. After many generations growing in the 15N medium, the nitrogenous base of the bacteria DNA were all labeled with heavy 15N. Then bacteria were switched to medium containing a light 14N isotopes and allowed to grow for several generations. DNA made after switch would have to be made up of 14N, as this would have been only nitrogen available and for the DNA synthesis. Melson and Sattel knew how often E. coli cells divided, so they were able to collect small sample in each generation and extract and purify the DNA. Then they measured the density of the DNA and density of density of DNA using density gradient centrifugation. Here is now this method separates molecules such as DNA into bands by spinning them at high speed in the presence of another molecule such as cesium chloride that form density gradient from the top to the bottom. The spinning tube density gradient centrifugation allow very small differences like those between 15N minus and 15N labeled DNA to be detected. Overview: the utilization of isotopes labeled to differentiate between parent and daughter DNA. Choose 40N common lighter and 15N rate heavier peak element of DNA. Technique of cesium chloride equivalent density gain centrifugation, separation of molecules based on density. Goal detect whether new nitrogen atoms appear on one or both daughter strands. So the result of experiments when the DNA result of experiment DNA made up of only 15 N form a single band. First replication band located at midpoint between 15 N and 14 N. Second generation had two bands, one at midpoint and one at 14 N. Same two bands sequenced subsequently. Uh, so now, the semi-conservative replication. As you can see, generation zero shows generation one shows DNA isolated after one generation. Also produce a single band where centrifuge. However, this band was higher intermediate in density between the heavy 15N DNA and the light 14N DNA. The intermediate band told Wilson and Sattel that DNA molecules made in the first round of replication was a hybrid of light and heavy DNA. This result fit with the dispersive and semi-conservative model, but not with the conservative model. The conservative model would be have predicted two distinct bands in this generation. And now second generation, the information from the second generation let Melson and Sattel determine which of the remaining models was actually correct. When second generation DNA centrifuge, it produced two bands. One was in the same position as the intermediate band and from the first generation, while the second was higher. These results told Melson and Sutter that the DNA was being replicated semi-conservative. The pattern of two distinct bands, one at the position of hybrid molecules and one at the position of light molecules, is just what we had expect from semi-conservative replication. In contrast, in dispersive replication, all molecules should have base of old and new DNA, making it possible to get purely light molecule. Now, third and fourth generation in the semi-conservative model, each hybrid DNA molecules from the second generation would expected to give rise to hybrid molecule and a light molecule in the third generation, while each light DNA molecules would only yield more light molecules. Thus, over the third and fourth generation, we had expect the hybrid band become progressively fainter, progressively fainter. As we can see in the figure, Mason Satter saw thus this pattern in this result confirming semi-conservative replication model. Contributor of biotechnology, most beautiful experiment forming prediction based on various models and pinpointing which matches experiment data address all models simultaneously, demonstrate effective unique mode of isotopes visualization, labeling not based on radioactive but rather density. Heavy isotopes are now widely used to label and analyze molecules, advancing the field of molecular biology. Semi-conservative DNA replication has been confirmed in every other study species. Understanding semi-conservative DNA is critical to elucidating functionally of cells of an organism. Further knowledge of mechanism of DNA and its interaction with enzymes, having a grasp on proper DNA replication allows for an identification of abnormalities, underlying defects, insight into heredity. सर हो गया. What sir? Hello. Don't know sir. Sir, हाँ sir, equalize isotopsin. Say 1 minute. सर ई कोलाई को लेके डिफरेंशिएट की मतलब हाँ ये हेलो
Yes, sir. Okay, sir. Okay, sir. Good evening, sir. My name is Vaishali Yadav, and my roll number is 35. My topic is replicating the end of chromosome. The end replication problem. Unlike bacterial chromosome, the chromosome of eukaryotes are linear, broad-shaped, meaning that they have ends. This end poses a problem for DNA replication. The DNA at the very end of chromosome cannot be fully copied in each round of replication because DNA polymerase can synthesize new DNA only by extending a primer. No doubt, DNA polymerase is not capable of completely copy DNA at the every end of chromosome. Therefore, approximately 50 nucleotides are lost during each cell cycle, which result in a gradually telomere length shortening. There is a special problem in replicating the ends, the telomeres of eukaryotic chromosome. A special mechanism is used for replicating the end of chromosome. Elizabeth Blackburn and Carol W. Grider have shown that an enzyme called telomerase. What is telomerase? Telomeric sequence is maintained by a special enzyme called telomerase. Telomerase, also called terminal transferase, is a ribonucleoprotein that adds a species-dependent telomerase repeat sequence to the third prime end of telomeres. Characteristic of telomerase. Telomerase is made up of both protein and RNA. It is a reverse transcription responsible for maintenance of the length of telomeres. Activity exhibited in gametes and stem and tumor cell. How telomeres work? Here is a diagram. After semi-conservative replication, the RNA remove and at the RNA remove and 3 prime and as a over left at a as a overhang, then telomerase bind to 3 prime overhang of the telomeres, which is complementary to the telomerase RNA. Then because are at using the telomerase RNA as a template, telomerase shift forward binding to the newly added repeat. Bases are added, telomerase shifted forward again, and the process repeat. DNA polymerase extend and RNA primer to synthesize a complementary strand. Here the written. Thank you. Yes, sir. Sorry. Germline cell and tumor cell. No. Because in prokaryote, it replication means prokaryote में circular होता है replication. नहीं, वहाँ पे requirement नहीं है. Uh, telomere repeat 2 times. Thank you, sir.
Yes, sir, I have sent you PPT. सर आपको सेंड किया व्हाट्सएप पे. Hello everyone, my name is Omkar Raute and I am going to explain the topic assembling the newly replicated DNA. So first of all, we are going to start from what is DNA replication. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. This is essential for cell division during growth and repair of damaged tissue. Assembly of newly replicated DNA into nucleosome in the cell of higher eukaryotes. The genome DNA, which is of a few meter in length, is compacted in such way as to be confined with a nucleus only micrometers in diameters. This impressive compaction is brought by association of the DNA with nuclear protein, on of which histones are the most prevalent. The resulting complex nucleoprotein structure is known as chromatin, and the basic unit of chromatin is the nucleosome core particle. This particle comprises 146 bp of DNA wrapped in nearly 2 tons, around an octamer of histone proteins made up of two copies of each of the core histones H2A, H2B, H3, and H4. So next slide. During S phase of the cell division cycle, not only is the entire genome DNA replicated, but the underlying chromatin structure has to be duplicated as well. It is widely assumed that the chromatin structure is transiently disrupted during passage of replication fork, and that new nucleosome are subsequently assembled on the emerging DNA daughter strand. So next slide. For replication to proceed, nucleosome must disassemble during the short time when the replication fork passes the newly replicated DNA into nucleosome almost immediately. The The new nucleosome are assembled as follows: each parental histone core of a nucleosome separates into 3-4 tetramer, two copies each of 3 H3 and H4, and two copies of an H2A H2B dimer. The next slide. The H3H4 tetramer is transferred directly to one of the two replicated DNA. Double helices pass the fork where it begins nucleosome assembly. The H2A and H2B dimers are released, adding to the pool of newly synthesized H2A H2B dimers. A pool of new H3H4 tetramers is also present, and one of these tetramers initiate nucleosome assembly on the other DNA double helices past the fork. So next slide. सर थोड़ा सा ऊपर सर. Rest of the new nucleosome are assembled from H2A H2B dimers, which may be parental or new. Thus, a new nucleosome will have either a parental or new H2 H3 H4 tetramer and a pair of H2A H2B dimers that may be parental or parental new. Histone chaperone proteins in the nucleus direct the next slide. Direct the process of nucleus assembly. As you can see in this diagram, the yellow color is the old stone. Red color is also the old stone, which is H2B, and dark blue is also the old stones, which is blue in color. And H4 is also the old histone, which is in dark green color. So we are going to see the new histone. The new histones are uh, H2A, H2B, H3, and H4. Thank you, sir. सर ये डायग्राम में सर H2A H2B डाइमर जनरेट होता है. टेट्रामर सर H2A H2B डाइमर जनरेट होता है. सर मोस्टली सर ओल्ड वाले डाइमर जनरेट होते हैं. हाँ सर होते हैं. आई डोंट. लिंकर हिस्टोन. सर H1 हिस्टोन. H1 हिस्टोन सर. सर हिस्टोन्स में लाइसिन लाइसिन अमाइनो एसिड, arginine अमाइनो एसिड. थैंक यू सर.
Thank you, sir. Thank you, sir.